Hello friends, today we are going to study about composition, structure and function of carbohydrates part 6. In this part, we'll study about the structural heteropolysaccharide. Now heteropolysaccharide, it is mainly function as an extracellular matrix, giving extracellular support to our cells. Apart from this, in case of bacteria and algae, they also form their cell wall, whereas in higher organism, they give extracellular support to the cells. Now for bacterial cell wall. Bacterial cell wall is a heteropolymer of N-acetylglucosamine and N-acetylmuramic acid which are linked by beta-1,4 glycosidic bond. This is how the structure looks like. One molecule of N-acetylmuramic acid is linked with another molecule of N-acetylglucosamine. The blue one is N-acetylmuramic acid molecule. The orange one is N-acetylglucosamine molecule. So alternatively, these are present. And one molecule of N-acetylglucosamine is linked with the next molecule of N-acetylmuramic acid through beta-1,4 glycosidic bond. Now, these straight chain polymer is cross-linked with the next straight chain polymer. And how they are cross-linked? If we see here, N-acetylmuramic acid of one chain is linked to the N-acetylmuramic acid of the another chain. But how? The first molecule of the first chain is linked with the third molecule of the next chain. These balls, they consist of amino acid in a sequence L-alanine, D-glutamate, L-lysine and D-alanine. So these amino acids are these four balls in a sequence L-alanine, D-glutamate, L-lysine and D-alanine. Apart from that, the chains are also cross-linked through this light colored balls. How many balls are present? 1, 2, 3, 4 and 5. So this is pentaglycine cross-link. So bacterial cell wall is a heteropolymer consisting of N-acetylglucosamine and N-acetylmuramic acid alternatively. Now, one molecule of N-acetylglucosamine is linked to the next subsequent molecule of N-acetylglucosamine through beta-1,4 glycosidic bond. Now, these polymer chains are placed side by side and they are cross-linked with small peptides. What are the linkages present? The N-acetylmuramic acid of one chain is linked to the N-acetylmuramic acid of the next chain, and the linkage is between first molecule with the third molecule. First molecule of first chain with the third molecule of the second chain. These amino acids are also cross-linked through pentaglycine crosslinks. 5 glycine molecules placed side by side. So this whole heteropolymer, because of this excessive cross-linking, they give a very strong sheath which surround the bacterial cell. And what function they play there? What role they play there? Protection. Protection of the bacterial cells from osmosis and from other organisms. So this bacterial cell wall provides protection to the bacteria. Because of this cross-linking, they form a very strong sheath and surround the whole cell, because of which it provides protection to the bacterial cell from osmosis and other organisms. The cell wall can be degraded by first lysozyme. It is present in our tears. It can degrade beta-1,4 glycosidic bond. So our tears or this lubricating agent present in our eye protects our eyes from the bacterial infection. Next is bacteriophages. What are bacteriophages? Phages means viruses. Bacterio means bacteria. So viruses that feed on bacteria or the viruses that kill bacteria are known as bacteriophages. Now, if you have studied about the life cycle of bacteriophages, when we study the microbiology. So if you see the life cycle of a bacteriophages overall view, then we know a bacteria attacked by a virus. Now, this virus will complete its full life cycle inside the bacteria and it will burst the bacteria and will come out. So, to come out from the bacteria, it should degrade the beta-1,4 glycosidic bond. If it degrades the beta-1,4 glycosidic bond, the cell wall will break open and the phages will release out. So, bacteriophages also have the capacity to degrade this bacterial cell wall, because until and unless they degrade the cell wall, they cannot come out and they cannot complete their life cycle. So bacteriophages can also degrade the bacterial cell wall, which is made up of N-acetylglucosamine and N-acetylmuramic residue. Next is penicillin. It actually don't break it. What its function is, it does not allow the synthesis of cross-linking, and that's how it prevents bacterial growth. So penicillin is the antibiotic which kills bacteria. How? Because they prevent the synthesis of this cross-linking. Okay, so now comes to algal cell wall. Now in case of red algae, the cell wall contains agar. This agar is made up of agarose and agaropectin. If we talk about agarose, agarose is a sulfated heteropolysaccharide. It consists of alternative unit of D-galactose and L-galactose. Here is the structure of agarose containing D-galactose and L-galactose. But here it is sulfated. You can see the sulfur group here. And also one more linkage you can see here is the linkage between C3 and C6 and ether linkage. So agarose consists of D and L galactose, where the galactose is sulfated and we can see the bond between C3 and C6 of same galactose. Now, what is the function of agar or agarose in the laboratory? What is its usefulness? Before studying about its usefulness, let's study what property of agar make it useful. So we know that we can dissolve agar or agarose in water by heating. So once we heat, the agarose will get dissolved in water, and when we cool down, it forms a gel-like structure. Now why it forms a gel, right? Because this agarose form double helix. Here is a chain. Suppose this is one agarose chain, this is one agarose chain. They will twist in such a way that they will form like this a helix having 3 residues per turn. Now inside this helix, they can fit water molecule. Water can get trapped. Now this one molecule, then again next molecule, then again next molecule, like this they can form a super molecule where multiple water molecules get trapped inside. And because of this property, they can form a gel-like medium. And because they can form a gel-like inert medium, hence they are used in first electrophoresis. You all might have heard about agarose gel electrophoresis, right? For the separation of nucleic acid. So here purified agarose is used as an inert base to run the nucleic acid. The second is you also might have used NAM if you are a microbiology student, that is nutrient agar media. What the function of agar here is also the solidification. One more function of agar is it used as a capsule to trap vitamins and drugs. Because these are inert, so they are used to trap vitamins and drugs, and we can easily consume it. So these are the importance of or uses of agar or agarose in a practical life, its application part. Now one question from my end for you all. What would have happened if bacterial cell wall is stabilized by alpha 1,4 glycosidic bond instead of beta 1,4 glycosidic bond? What would have happened if bacterial cell wall is stabilized by alpha 1,4 glycosidic bond? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.